Hello, and welcome to another episode of the Med School Minutes podcast, where we discuss what it takes to attend and successfully complete a medical program. This show is brought to you by St. James School of Medicine. Here is your host, Kaushik Gua.
Thank you so much for joining us on another episode of Med School Minutes, where we talk about everything MD related with the focus on international students, specifically students from the Caribbean. Today we have a very special guest, Dr. Alya Limayem. She is an associate professor at the University of North Florida, and she specializes in nanotechnologies. Today we're going to delve into what exactly is nanotechnology, and we're talking to Dr. Limayem, who is a pioneer in this field with 3 patents to her name. Without further ado, let's welcome Dr. Limayem.
Thank you so much for joining us, Dr. Limayem. We really appreciate you taking the time out of your busy schedule and meeting us in beautiful and usually sunny St. Vincent. Today it's raining a little bit, but once again, thank you so much. We're honored to have you. Why don't we start by you giving us a little bit about your background and all the accolades that you won over the last couple of years.
All right. So first of all, thank you for having me. I'm so excited and honored to be here with you. Very much appreciated. I have to know St. James School of Medicine. It has lots of potential, and in the horizon, you can do many things to this school. So let me introduce myself by saying that I'm an associate professor. I obtained my PhD from the University of Arkansas, Fayetteville. And mostly I've been working on designing, manipulating nanocompounds to use them in order to increase productivity and enhance the yield from renewable energy. And by that time, the trend was yeast-based fermentation, the biofuel, the bioethanol. And from there, we have noticed that we have like some drug resistance because we're using antibiotics to remove the yeast, to remove the bacteria that were competing with the yeast in order to convert sugar into alcohol. Right. So we started to think about a new methodology using a non-traditional conventional antibiotics that are cost-effective, safe, and effective against some type of bacteria. And from here, where I get the idea to work with nanocompounds.
Okay, I see. So when we read your bio, it says that you've done a lot of research in nanotechnology. Can you explain what that really is?
So the nanotechnology is the science that manipulate and design biological matter for my field. It can work, of course, for mechanic or electronic, but my kids manipulating this teeny tiny, we will say from 1 to 100 nanometers, which is billions meter. And I have noticed on them their amazing properties for precision. And that and we can find some of them can work like antimicrobials, very cost-effective antimicrobials, and that can be potent if they're selected in a way and designed in a way that can target, reach the target cells that we need.
Okay, awesome. So right now, what does your research largely entail? What are you doing?
So my research is mostly with drug. I started with drug resistance because the emerging drug resistance worldwide, not only in the US, everywhere. We've noticed that almost 1 million, more than 1 million deaths per year, and associated to many hospitalizations that can reach millions. So this was alarming, and we need really to think about a sustainable solution that could protect us from this emerging. So the conventional, like I have noticed 10 years ago that you are reaching a pipeline of drug resistance. So we have bacteria that are resistant to 10, 12 antibiotics. And many immunocompromised population, especially with the hematological malignancies or any patient with impaired immune system can actually can pass away from drug resistance infection.
Okay.
So the nanocomponent can be the first alternative to this type of hospitalization disease.
And if I'm not mistaken, you have a couple of patents to your name. Can you talk a little bit about those?
Of course. So especially the most important one is the nano formulation, including zinc oxide and chitosan. So I selected them for their effectiveness, potency, and together they're broad synergistic and broad spectrum against both gram-negative and gram-positive bacteria, because we have wide range of drug-resistant bacteria, especially the hospital-acquired one. We call them the nosocomial pathogens. So these two nanocomponents are very effective, and we have evidence for that. And also, they can be used as a supplement, not only as antimicrobials.
And how do you administer these nanoparticles into the human body?
So depending on where you will administer it, so it can be, we talk about nanostructures. So the nanostructure, if it's, for example, for the skin, and I believe here, for example, in St. Vincent of the Caribbean, it's very important to look at the type of nanocompound that treat the skin for the waterborne pathogens and mosquito, etc., bites. So that can, if it's for the skin, the liposome nanostructure is perfect. Because it's site-specific. If it's going to be ingested, the nanomicellar form is the most appropriate. First, for its size, because if it's more than 200 nanometers, can be cleared by the spleen or by the liver, and if it's very small, it can be cleared or eliminated by the kidney. So we want it to be in between 100 to 150 nanometer. Then we can warrant its bioavailability and have it reach the target cells in the form of nanomicelle. Now, if you want to use it like a spray or an inhaler, if you want it to be inhaled, that microsphere with a size of 2 micrometer, says 2000 nanometer, is fine, as long as we can also embed it within, embed the zinc oxide and the chitosan inside this microsphere, so it can reach the lung, and we can have the effect we need.
And have you done any research about the exact structure of the nanosphere?
Oh, of course, absolutely. So I started with the nanomicelle because we wanted to be ingested and compare the ingested to the inhaled. And we figured out that the core of a nanomicelle is hydrophobic. It means water repellent. So you had to design hydrophobic zinc oxide to have it there. And warrant the zeta potential and the polydispersity index conforming to our needs. So this is overall. And for the microsphere, we bought a machine we call spray dryer. And these days with my team, we're trying to figure out what would be the best size versus make it flow and effective as much as we need.
Which one of, so you have 3 patents. Which one of these patents do you think is closest to real world deployment?
The one that can be used to treat water surfaces. So I received grant for environmental pollution commission, and we really figured out the potential of the zinc oxide and chitosan together. We call it CZNPS together, and they're very effective against the nosocomial pathogens, since the water can be mixed with not only the household, but the waste from pharmaceuticals, and hospitals, etc. So we really figured out the effectiveness of these nanocomponents against these nosocomial bacteria. Use it for reclaimed water.
So one question is, I know we've talked about, and you mentioned this, the West Africa study that you've done on indigenous
West
therapy. how would these differ from off-the-shelf probiotics, really?
Yeah, here the indigenous, or we call them um, bacteria or probiotics. We can talk about bacteriotherapy, which is a new technique.
Right.
But the good thing is they're used to the diet of the locals
Okay.
and the immune system.
Okay.
So they are more effective than the off-the-shelf generic
I see.
probiotics.
Okay. I see.
And we had so many different experiments with them. They did it in situ in their countries,
Right.
and they figure out the effectiveness
Okay.
of the local versus indigenous
Right.
when compared to the generic one. They have the competitive exclusion, for example, with the pathogens
Right.
is better, and they have better results and outcomes.
Okay. So now switching gears a little bit, going for your day-to-day responsibilities of, as you mentioned, you are an associate professor at University of North Florida. How do you balance your time between teaching and the research that you have to do? Because I'm sure it is not a 40-hour work week for sure, or is it?
It depends.
Okay.
I have some intensive days, especially during fall and spring.
Okay.
I have to work hard with the students.
Okay.
It's not just teaching itself, because I enjoy teaching.
Okay.
I giving messages, teaching new concepts to my students, giving them ownership of their new studies, etc. But the most challenging part is when the 150 students want to reach me in person every day.
Right.
So 150 together, answer to their questions, the one I dream can be sometimes challenging when you have establishing new labs.
Right.
And want to think more about the research projects, and you progress
Right.
in that sense.
Okay.
But in any ways, I always try to find to set boundaries when necessary,
Okay.
and I maintain my balance by doing sports, going to the gym. I like dance, tennis, and sometimes have break with my friends and family. So this will give me type of energy, positive energy, and feel rejuvenated and refreshed
Right. Right.
to restart my day.
Right. So what's your average work week like? Is it 40 hours, 60 hours, 80 hours, more than that, typically?
I will say 50.
50 hours.
Yeah, around 50 hours.
And that includes teaching, classroom, as well as handling student questions, and grading papers, etc., and course responsibility, as well as your research.
And in addition, I'm a first lady. I should say this.
Okay.
And being a first lady, I do my best to promote UNF, and I believe also UNF is a very promising, excellent university.
Absolutely.
It has a lot of potential,
Right.
and I really believe in its success if we all work together and use its potential in that sense.
So here's a question. I mean, obviously, St. James is an offshore medical school, and traditionally, offshore medical schools haven't or don't do a lot of research.
Mm-hmm.
As a school, as a Caribbean school, or as an offshore US-based Caribbean school, if you will, St. James has a lot more research requirements and focus on research than most other schools. That being said, that is significantly less than most research universities in the United States. How would you suggest a school like St. James can potentially level up its research, if you will?
Well, very important things, and a very excellent question, I believe, and authenticity does matter.
Okay.
So its place, it's unique.
Okay.
Don't try to mimic others, but be yourself.
Okay.
And I believe here in the Caribbeans, you have the marine microbiota.
Mm-hmm.
You do have also the waterborne pathogens, and all these type of things can be enhanced. It's underexplored, I would say, and you can further explore the studies that belongs, make a niche, create a niche that belongs specifically to St. James School.
Okay.
So by building your uniqueness
Okay.
and your and focus on your authenticity,
Right.
you will stand
Right.
above
So-
the others.
So, in your opinion, do you think a simple wet lab that is typically available in most schools is adequate enough to get, say, groundbreaking research and potentially getting patents and grants like you've done?
To be honest with you, I started my career with a small startup.
Okay.
Okay, so
Okay.
that means that said, where there is a will,
Okay.
there is a way.
Awesome.
So you need to have a realistic idea
Okay.
that is plausible,
Okay.
not something for the prestige or
Right.
over ambitious ideas or
Okay.
something that does not fit your resources
Okay.
and fit the context and the culture in place.
Okay.
So what we need, for example, when I started my studies, I have plates, media,
Yeah.
and in my mind, how can I get rid of drug resistance, which
Right.
is true problems,
Right.
which is significant.
Right.
And I started the beginning using the plate and the drug and assessments against the colonies.
Okay.
And suddenly I saw that this drug was able to inhibit
Okay.
these colonies, and
Okay.
it costed for me.
Okay.
It was very cost effective. Doesn't need that much money. So
Okay.
this I could use like preliminary data.
Okay.
And it was successful. My grant was accepted based on very small.
Right.
And I was not even starting like a research professor, research assistant professor. I was not even allowed to have PhD student with me.
Okay.
So I started with undergraduates,
Okay.
and I was able to be scored from the NIH, and that was my beginning. Because the idea was very plausible. With collaboration, tried to collaborate with senior professor who kindly, I remember by that time, Dr. Lancy Shaw, he kindly gave me his set of drug-resistant bacteria, received it from Moffitt Cancer Center. And from there, I started my experiment. So, and they were not costly, but with collaboration, I was able to build my own world.
I see. Wow, that's really interesting.
Yeah.
So obviously, whenever we talk about research, a lot of people will say that research is all about failing and not giving up. In your experience, what is a big research push that you've made that wasn't very successful, but you learned a lot from?
I mean, I was fortunate.
Okay.
Everything. I mean, at the beginning, I have to be honest with you, not so many that believed in me.
Okay.
That was the most challenging part.
Okay.
Even saw people laughing, etc.
Okay.
But I did not pay attention to this.
Okay.
I persevered
Okay.
and continued to be innovative. It's like
Okay.
the perseverance with the creativity and innovation,
Right.
not staying just taking one way. No,
Okay.
it's this way. If it does not work, I take another way.
Okay.
I take another approach,
Okay.
another methodology. Look for more matching collaborators.
Okay.
So perseverance with creativity and innovation will help move forward.
That's awesome. That's actually really good advice. I mean, as a school, we are definitely trying to improve our research capabilities. And obviously, with your guidance and your help and mentorship, I think that will definitely happen. But I do want to take a step back and go back to your very beginning, because you originally are from Tunisia.
But my kids are American, and we are well established that American. When I visit Tunisia, nobody recognizes me now.
Oh.
I changed.
Well, I'm pretty sure with all the patents and all the coverage that you've gotten in the last couple of years, I think that will change very soon. People all over the world will start recognizing you.
Okay.
But I definitely wanted to know, when you came to the US, what was the journey like? Did you come with the intention of being a trailblazing scientist? Or was it that, hey, you know what? I'm just going to make my way and let's see what happens? And
the second one.
Okay.
Oh, the second one. I came pregnant when I came here
Okay.
with Sarah, and I want to succeed. Okay, and did all my best to succeed. And it turned to be inventions and top 3 papers.
Awesome.
Yeah.
Awesome. And so the field that you eventually got are in today,
Yeah.
nanotechnology, is this something that was well-intentioned right from a very early age, or when you were young, were you dreaming of being a pilot, for example? I mean, all of us in prep there completely.
No, it came like this. I like science. Okay. I'm very curious and resilient person. I came also from a curious family. My dad is an engineer. Okay, he built his own company in cheese and making. My mom also studied biology and she's very gifted for art, etc. So she involved me always. So coming from a family that they have these orientations also gave me the taste, and no wonder that I'm also biologist and I like art and move forward with all these gifts from.
Now I'm beginning to see it, because cheese definitely needs a lot of microorganisms and a lot of fermentation processes. So I can see where that interest probably originated.
And then they're beneficial, beneficial for health, but also delicious when we eat them.
Right. And eventually, when you moved here and you started your research, as you said, you obviously you started with one aspect, and then you had a eureka moment, right? That says, "Oh wow, maybe this is something I need to focus." Can you describe when that eureka moment really happened, that you decided that you want to focus on drug resistance?
Yeah, it happened to me when I realized the impact of drug resistance, not only in the US, but worldwide.
Right.
Yeah. And then I said, why don't we look for another alternative that could be effective and will somehow diminish this pipeline drug resistance? And Dr. Ricky, my advisor over there, Stephen Ricky in Arkansas, also encouraged me to take this direction, and also offered to me the resources and the necessary knowledge for that. So I used my curiosity and perseverance to take this direction, and found myself with many discoveries, inventions, and original contributions.
That's awesome. So, in general, from a macroeconomic standpoint, not economic, but macro healthcare standpoint, what is your general view on countries that don't really seem to pay a lot of attention to drug resistance? For example, in St. Vincent, you can walk into a drugstore and get non-prescription antibiotics.
Yeah.
In India, I mean, technically you need a prescription, but anything goes, kind of a situation. What are your views on that? And I believe, if I'm not mistaken, I think the World Health Organization has also shown concern, especially with India's population, that drug resistance could potentially be catastrophic.
So we need to spread awareness. And tell them, look at the pandemic. Because I personally was involved also. I received the grant for the rapid response, COVID-19. And zinc oxide also turned to be effective against viruses like COVID that have envelope. So it can destroy its envelope and the genomic material and the protein spikes. It has an effect on it. And it can be removed, can be used in wastewater, reclaimed water, and then recovered. And we can have it with the membrane filtration. So we recover the water from all chemicals and So this can happen. So by spreading awareness, then look at what teeny tiny small organism like corona. Did not pay attention to it. We need it, and it impacted the entire health system and worldwide health. So drug resistance could be also an invisible pandemic. And unfortunately, we can lose people that from our families, or if they are immunocompromised or they gain age, elderly, that can be impacted and affected by this. So think about them.
But what are your thoughts on a lot of people including myself, when I am not feeling very well and over-the-counter drugs aren't really helping in a day, maybe a day and a half, I'm like, you know what? I went to St. Vincent. I'm going to pick up, I always pick up antibiotics and I just take them. And I go through the course as recommended on the box. What would you say to people like that?
I will tell them there are not only antibiotics that are effective against this type of diseases. And they certainly know, for example, honey, ginger, turmeric.
Right.
Turmeric and ginger. Start with those first. Start with a nutraceutical. Drink lots of fluids, make soups with onion and garlic. These are very effective and sometimes are better than antibiotics. And add to them when you eat at some time, rosemary. So start with a natural herbal extract and spices. Be patient for a couple of days and have some Tylenol if you want, or to release the pain. And you will see. It's going to, I mean, if it's viral, look at your, the discharge should be transparent. So you don't need antibiotic at all. So by consuming antibiotics, you are increasing and exacerbating the drug resistance. That's what you're doing. In your body. And if it's greenish and it continues after a few days with high fever, then, and you consume some of these herbal teas, turmeric, ginger, lime, etc., then after 3 days, you go and check the doctor for antibiotics.
That's very interesting because, you know, my knee-jerk reaction always is that if I'm feeling sick, I just don't have the patience, you know, and I'll just go, oh, there's antibiotics. Let me just pop in and use it. And I don't know, it might be psychosomatic, where I was just all of a sudden cured.
Just think antibiotics are only effective against bacteria and specific type of bacteria. So you might does nothing but exacerbating drug resistance. And also affecting the microbiome. Kill the good ones that helps you enhance your immune system, enhance digestion, correlate with the brain and lung, etc.
So, like my final question really is, for a school like St. James, we have basic research where our students, every student attempts to get published, and every year I think we get about 200 to 400 publications, some of them in quite prestigious journals as well. For a school like ours, and a lot of our publications also come through our hospital partners as well. But for a school like ours, we're trying to develop the research capabilities. What do you think should be the very first step? We have obviously wet labs and things like that, but what do you think should our very first step be?
First step, look at the problem. What is the problem? Its significance. Study it. We have to measure it and have real data that measure the problem and the significance of that. Then look at collaborators, national and also international, and this will help. Then make sure that we have enough resources. And this resource will fit the context and the culture in place. Like as I mentioned previously, that you look at the microbiota in marine systems, skin's problem, waterborne, the virus like Zika virus can be a potential subject. Like I attended conferences and I saw that now they're thinking removing breeding sites of certain mosquitoes, like the Aedes aegyptis, it's called like that. And if you remove these breeding sites, you can actually diminish and reduce the occurrences of many different diseases. This is a very plausible project that you can stand above with your uniqueness and continue creating and helping student success. So this also will promote St. James School of Medicine for research and everything, and create a new pattern.
Excellent. Well, thank you so much for your time.
My pleasure. Pleasure was mine. Thank you for inviting me
Oh, it's our
to
pleasure.
share my knowledge.
Yes. It's been a pleasure to have you. Our students were very encouraged by your talk yesterday. And I hope the feedback that you're taking back with you about our students is very favorable. And obviously, there will be more discussions as the week goes along. But thank you so much for your time.
The pleasure was mine. Thank you so much.
Thank you so much for joining us on this very inspirational episode of Med School Minutes. We learned a lot about what nanotherapeutics is and where the future of healthcare really resides. Thank you so much, Dr. Alya Limayem, for taking the time out of your busy schedule to visit us in sunny St. Vincent. And remember, if you really enjoyed the contents of this podcast, please download, follow, share, and like our podcast. It goes a long way for our team. And remember, there's no shortcut to becoming an MD.
Thank you so much for tuning into our show. We hope you enjoyed another episode of Med School Minutes. If you like our content, please follow us and receive notification when a new show is posted. This podcast is brought to you by St. James School of Medicine. For a video version of this podcast, please check us out on sjsm.org/video.